Hello everyone, my name is Tristan, and in this video I am going over the proximal convoluted tubule. Alright, so nephron has the renal corpuscle, which is the glomerulus and Bowman's capsule, and then after this renal corpuscle, after we leave this Bowman space, we go into the first part of the nephron's tubular structure, which is the proximal convoluted tubule. All right, what we see right here is the proximal convoluted tubule. Here is the tubular lumen, right? Here are the tubular cells, the boxes, and outside of these tubular cells are the capillaries around it, the paratubular capillaries. All right. Now, this membrane here is the basolateral membrane. All right, the membrane that is facing the paratubular capillaries. The membrane facing the lumen of the proximal convoluted tubule is the luminal membrane. Okay, straightforward. So we have the lumen, the tubular cells, and the paratubular capillaries. All right. So the glomerulus brought a bunch of stuff through its four barriers into Bowman space. It brought glucose, amino acids, electrolytes like sodium, potassium, magnesium, calcium. It brought certain lipids. Maybe it brought small proteins in. It brought a bunch of stuff in, right? And the proximal convoluted tube's job is to do two main things, all right? And these two main things are reabsorption and secretion. So in a nutshell, this proximal convoluted tubule, or PCT, can reabsorb stuff, meaning that it brings stuff from tubule to bloodstream, right? Reabsorbing things back into the bloodstream. A bunch of stuff got into the tubular structure via the glomerulus. It's inside, but the body wants some of that stuff back, right? Because we just brought a ton of stuff in, but some of that stuff is important and we still need it for the body, right? We don't want to excrete it as urine. So what are we going to do? We're going to reabsorb it back into the bloodstream so the body can utilize it. Secretion, on the other hand, is bringing stuff into the tubules, all right? So this is bloodstream to tubule, all right? Now an example of this could be things like drugs, right? We want to bring drugs into the tubular lumen so that it can be excreted as waste, right? Another example is uric acid, right? These are the things that are secreted from the bloodstream to the tubular lumen, all right? So, let us first talk about glucose and amino acids. So, erase this. With glucose and amino acids, they are important, right? They're important, and we don't want to excrete them as urine, right? After the tubular structure of the nephron, the filtrate goes down through the ureters and becomes urine and gets released via our urethra, right? So we don't want certain things like glucose and amino acid to leave the body. So what are we going to do in this case? We talked about that the PCT had two functions, reabsorption and secretion. What are we going to do with glucose and amino acids if it's important for the body? We are going to reabsorb it from the tubules back into the bloodstream. How does that occur? right? So we have glucose that got through the glomerular membrane and the rest of the barriers. We also have amino acids, right? They're in the Bowman space, now they're going down through the PCT. All right, here's what happens. There is a transporter here. This transporter is present on which membrane, right? It's present on the basolateral membrane. That is the membrane facing the peritubular capillaries. So this basolateral membrane has a certain transporter, okay? This transporter is a sodium potassium ATPase, right? So what does it do? It brings in 2 potassium and takes out 3 sodiums, right? So 2 potassium go in to the tubular cell, and 3 sodiums leave the tubular cell into the interstitium and eventually the bloodstream. And this process requires ATP, all right? So this process needs ATP in order to occur, all right? The reason that the kidneys do this, the nephrons do this within the kidney, is it's very smart, right? We're taking sodium out. We're creating a concentration gradient, right? So this sodium, it's going against its concentration gradient. That's why it needs ATP, right? But because these ATPases are taking sodium out of the tubular cell, it is allowing, let me use a different marker. It is allowing another transporter to bring in sodium via passive diffusion, all right? So sodium in the tubular cell is leaving via active transport. It needs ATP, 2 potassiums are going in, and this is creating a concentration gradient so that the sodiums inside the tubular cell, the concentration of sodiums inside the tubular cell is less than the concentration inside the lumen of the proximal convoluted tubule. All right, so this is allowing sodium to come in to the tubular cell by a passive diffusion. Sodium's coming in, all right? Now, what this allows the kidneys to do is to bring various molecules in against the concentration gradient, right? What are some examples, right? We're talking about glucose and amino acids here, right? So as a consequence of sodium being passively diffused, that sodium that is being brought into the tubular cells are bringing along glucose and amino acids with it, all right? So with the sodium, we're bringing in glucose and amino acids, right? This transporter is bringing in sodium, and as a consequence of the sodium streaming in, it's bringing the glucose and amino acids along with it, so that these glucose and amino acids can eventually leave the tubular cell, leave the basolateral membrane, and get back into the bloodstream and be reabsorbed back into the bloodstream so it can be utilized by the body. All right, this is an example of secondary active transport. All right, secondary active transport. Why is it secondary? The kidneys are smart. They are utilizing ATP only to get sodium out of the tubular cell, to create a concentration gradient that allows the concentration of sodium to be less here compared to here. Because of that, it allows sodium to passively diffuse through the luminal membrane into the tubular cell. And because of that passive diffusion, it is assisting, it is bringing in other molecules along with it, right? Because sodium's everywhere, right? It's all throughout this PCT. And within the sodiums, there's also glucose and amino acids and things like lactate and such. So that when sodium is passively diffused goodness gracious, into the tubular cell, it is also bringing in the stuff that is around it, glucose, amino acids, right? That is secondary active transport. We are utilizing active transport here to create a concentration gradient so that we can bring in glucose and amino acids along with it. Boom. Let me pick up my markers now. So that, my friends, is secondary active transport. Let's talk about how bicarbonate is reabsorbed now. All right. So we talked about how glucose and amino acids are reabsorbed. What about bicarbonate? Okay. With glucose and amino acids, 100% of glucose and amino acids are reabsorbed. All right. So that means that the glucose and amino acids that pass through the glomerulus into Bowman space, 100% of those molecules get reabsorbed back into the bloodstream, right? Because they are useful. We need those. 100% reabsorbed. Let us move on to how bicarbonate is reabsorbed. That is important as well because bicarbonate is alkaline, okay? It's basic, all right? So by reabsorbing it back into the bloodstream, we make the pH of the blood more basic, making the pH of the filtrate that we are creating more acidic, right? We are throwing in hydrogen ions into the lumen and bringing back our bicarbonate to sustain the pH of our blood and get all that acidity out of the blood into our filtrates. All right, so let's get into that. All right, bicarbonate. CO2 comes in, right? Comes into the tubular cell. CO2 combines with hydrogen. All right, it combines with water. And this combination creates carbonic acid. So CH2O3, carbonic acid. Carbonic acid then dissociates into two things. It dissociates into bicarbonate, right? And hydrogen ions. All right? So CO2 and H2O combine with the help of the enzyme called carbonic anhydrase. Right? Let me write that down. A second. So CO2 and H2O combine. What helps this process? Carbonic anhydrase. This is an enzyme, all right? An important one. Carbonic anhydrase combines CO2 and H2O to create carbonic acid. Carbonic acid then dissociates into bicarbonate and hydrogen ions, all right? Now, there's a transporter, right? There's a transporter on which membrane is this? The luminal membrane, facing the lumen. There's a transporter on that luminal membrane, and this transporter brings in sodium, right? So it brings in sodium into the tubular cell, and this hydrogen gets released into the lumen, right? Via this transporter. And this transporter is what is known as an antiporter. And this process is also secondary active transport, right? Why is it secondary active transport? The same reason, right? We have another transporter. I draw my markers every 3 seconds. We have another transporter, same one as this one, that is bringing potassium in and getting sodium out via active transport, which means it needs ATP. Because of that, it is reducing the concentration of sodium inside of the tubular cell, allowing sodium to passively diffuse into that cell, right? That's why this is secondary active diffusion, right? So now sodium's coming in and it is assisting those hydrogen ions to get into that lumen, right? Secondary active transport. So hydrogen ions are now inside the lumen, all right? What is that doing? That is increasing the acidity of this filtrate that is being created here in the proximal convoluted tubule. Where does this bicarbonate go? It goes back into the bloodstream. It is indirectly reabsorbed, all right? So what happened? Carbon dioxide plus H2O combined with the help of carbonic anhydrase into carbonic acid. Carbonic acid inside of the tubular cell then dissociates into bicarbonate and hydrogen ions. Hydrogen ions get into the tubular cell via secondary active transport. Bicarbonate freely goes back into the bloodstream, gets reabsorbed, all right? That is how we reabsorb bicarbonate, thereby making the blood more basic, right? Bicarbonate is an alkaline molecule. Hydrogen ions create acidity, makes the filtrate more acidic. So by doing this process, by bringing in CO2 into the tubular cell, having it combined with hydrogen and doing all this good stuff, we are bringing in acidity out of the bloodstream into the lumen of the proximal convoluted tubule. This acidity then leaves the body as urine, okay? So that is bicarbonate. Now, how much bicarbonate is reabsorbed? Glucose and amino acids, 100% of those things are reabsorbed. Bicarbonate is 80 to 90% reabsorbed. Now remember, this reabsorption is occurring indirectly, okay? Right, because we have to bring CO2 in to create carbonic acid, then that carbonic acid will dissociate into bicarb, okay? So it's an indirect reabsorption. Now, let's talk about water, okay? Water and sodium, right? Let's talk about water and sodium. So H2O and Na, all right? Water is our universal solvent, all right? And this solvent likes to go where there are higher concentrations of solutes, okay? This process of moving towards an area where there is higher solute concentration as water, this process is called osmosis, okay? So let's go to this tubular cell here. This tubular cell, what do we have going on? Same thing, right? We have that sodium potassium ATPase bringing potassium in, taking sodium out, creating a why am I blanking out? It's creating a concentration gradient that is allowing sodium to passively diffuse, right? So sodium's passively diffusing through here, okay? Right, we got sodium. A bunch of it's coming in, right? Eventually, that sodium goes through the basolateral membrane into the bloodstream. We talked about osmosis, right? Osmosis is when water goes to areas where there are higher solute concentrations. It's going to follow this sodium, right? The sodium in the lumen. There's also, what's another component of plasma, right? Water. This water, this H2O, is going to follow this sodium. Where is it going to follow it? Back into the bloodstream, right? So that's an easy understanding of how sodium gets back and how water gets back, right? Wherever sodium goes, water is going to go as well. All right, because water likes its solutes, okay? So, what percent of water and sodium is reabsorbed? Around 70% reabsorption. Now let's talk about why water and sodium reabsorption is the same, right? 70% of sodium gets reabsorbed, so 70% of water is going to get reabsorbed as well. Because why is water able to get back into the bloodstream? Because sodium's getting back into the bloodstream. Wherever sodium goes, water goes. What is this process called? Osmosis, all right? Now let's talk about the other electrolytes, like potassium and calcium and chloride, right? So what have I drawn here? I've emphasized that there are these tight junctions in between the tubular cells. There's spaces in between the tubular cells, and these things, these electrolytes, calcium, chloride, potassium, right? They can move in between these tubular cells and get reabsorbed. So they move in between the cells, get reabsorbed, right? They move past these tight junctions and get out of this lumen, right? Electrolytes are important as well, so we need to be reabsorbing these as well, all right? What is this called? What is this process called? This process of moving in between these tubular cells is paracellular transport. Paracellular transport. All right. Now, paracellular transport is a passive process, okay? There's no ATP required to bring these electrolytes out of the lumen of the tubules into the bloodstream, all right? Now, what we've been talking about here with glucose and amino acids, as well as carbonic anhydrase, as well as the movement of water and sodium into the bloodstream, these three processes are transcellular transport, okay? So this right here is transcellular, meaning that you're going through the cell, not around it. You're going right through it, all right? Transcellular transport, paracellular transport, okay? So with lipids, lipids are very easy to reabsorb. They are also passive transport, and they are easy to reabsorb because they are lipid, okay? They are fat soluble, meaning that they can easily pass through phospholipid membranes, right? We got a phospholipid membrane here, the luminal membrane, and the basolateral membrane, also phospholipid, right? So these lipids, they can just pass through. Piece of cake, okay? That's really all I got to say about lipids. Small proteins, if there are proteins present inside of the lumen, there are these receptors here. There are these receptors. As the proteins go down to the proximal convoluted tubule, these receptors grab on to the proteins, right? Maybe insulin, maybe hemoglobin, right? Hemoglobin. These receptors grab onto it and perform what is known as receptor-mediated endocytosis. Receptor-mediated endocytosis. Okay? Receptors grab onto proteins, endocytosis, engulf into the tubular cell. Then there is a vesicle with the proteins inside as well as the receptors, right? And what happens is that there are a bunch of hydrolytic enzymes that come in and destroy these proteins. They degrade the proteins. They hydrolyze the proteins, turning them into their monomers. What is the monomer of protein? Amino acids, right? So this turns into amino acids, right? Beautiful. Now, one more thing. Secretions, all right? Say we have drugs, okay? We got drugs inside of the bloodstream, right? It could be penicillin or morphine. We got drugs. And these drugs, they need to be taken out of the body, right? Maybe we can't properly filter them, right? With our liver. So we just got to take them out. These drugs in the bloodstream, we got to secrete them into the kidney tubule so they can be left, so they can exit the body via our urethra as urine, right? Drugs come in. Boom. What's another example? Other examples are bile salts or uric acid, okay? These things, they need to be secreted out of the bloodstream into the lumen of the tubule, into the proximal convoluted tubule. What is this process called? Secretion. Secretion is always active, okay? It is always an active process, meaning that it always needs ATP in order to occur. Reabsorption can involve active transport, but it does not always need active transport, all right? Things can diffuse passively into the bloodstream. Like with our electrolytes here, they are cellular transport. They do not require the use of ATP. They get into the bloodstream, get reabsorbed passively. Secretions always need ATP. They are always active, right? Why is this significant? It is significant because when we take drugs, when we continuously take drugs, we continuously utilize ATP and need to utilize ATP in order to filter these drugs out of our systems, okay? So if we have patients that have excessively taken drugs, right? Taken antibiotics. These antibiotics, like penicillin, they are considered nephrotoxic, okay? Because we're bringing them into the bloodstream, or we're bringing them into the proximal convoluted tubule, right? And this is requiring ATP, a lot of it, right? So this can, upon excessive, right, use of bringing drugs into the tubule, that can harm the kidneys, all right? So that is our proximal convoluted tubule. We went over how we brought in glucose and amino acids back into the bloodstream, how we bring in bicarbonate indirectly, how H2O and sodium get reabsorbed via osmosis, right? And then we talked about our cellular transport with our electrolytes, and we talked about secretions and how drugs, as well as bile salts and uric acid, need to be secreted and how that is always active transport, right? We also talked about our proteins, right? Our proteins, if there are any proteins, can be reabsorbed via receptor-mediated endocytosis, right? These receptors bring the stuff into the tubular cell, then a bunch of hydrolytic enzymes eat them up, mash them up into their monomers, which are amino acids. Those get reabsorbed. Boom, you got more amino acids into the bloodstream. So that is our proximal convoluted tubule of our nephron. Thank you for watching.